and welcome to Miss Estruch Biology. My name's Miss Estruch, and if you are new here, then I'm here to help you to get to grips with those challenging topics in biology, improve your study skills, techniques, and help you to get the grade that you deserve. And today's video is going to be, I hope, an absolute game changer for paper 3 on Friday for AQA Biology. Because you might have already seen my essay prediction video that I did about a month ago, and I've posted about it on Instagram and TikTok. But I have now had a look at paper 1 and paper 2, and I've come up with my scribbled notes where I've gone through what topics have come up that were on the list, that weren't on the list. I've looked at paper 3 to see what is still due to come up. I've then compared all of that to the specification, and I've picked out the 4 parts of the specification that haven't come up at all. And from that, as well as historic titles that used to be quite popular and haven't come up in a while, I've come up with these 3 essay titles. So stick around for this video. I'm going to go through each essay title and talk to you about a model essay plan. Now, if you do want even more help with your essays, then I've got a few things that could help you with just that. Number 1, if you haven't seen my 3 other videos that I have on YouTube for essays, I have one about how to write an essay. I have another on how to get full marks. And my 3rd one was marking the essays that people submitted to me. So definitely check that out for more help with the essay. Now, the essay, I cannot emphasize enough how important it is to prepare for this. It's 25 marks, and that is actually nearly 10% of your entire A-level grade. So it's really important that you do put the time in preparing for this essay. You should spend about 40 to 45 minutes on the essay because it is worth so many marks. And if you're struggling to be able to identify the linking points, which they say in the mark scheme is the interrelated points, then definitely check out my A-level notes, because for every single topic, I tell you exactly what you could link it to in terms of the importance of for the essay. So I'll put that in the description bar below so you can check those notes out. Oh, and as a side note, if you get the reference of my t-shirt, please comment below, because this is absolutely one of my favourite shows and I have loved the most recent series. So if you know what this series is, then oh my gosh, how amazing is it? But for now, let's think about essays, and we can watch Netflix and TV shows in about a week's time. So here are the 3 new essay predictions. I did have some older ones, but I've changed them after having a look through paper 1 and paper 2, looking at what is left to come up on the paper 3 advanced information, and then looking at the remaining topics. So I've based this on the topics that haven't come up really, or not at all, in the whole A-level and aren't expected to on paper 3, but are fundamental topics to A-level biology. And also linked to titles which are quite common and come up every few years. And I know straight away some people have already said to me the 3rd prediction was 2020, so surely that's too soon. Now that actually leads me to a bit of a disclaimer. With these essay predictions, number 1, it is just based on the research that I've said, so don't go in thinking it's definitely one of these 3 and I'm not going to do any other preparation. This is to give you an idea of what I think the titles might be similar to. I do not expect any of the titles are going to be word for word the same, but I would expect that maybe at least one will have some similar concept. So there might be one that is linked to DNA or microorganisms or bacteria. There might be one linked to variation and diversity. Even though it came up in 2020, there's not been really much on it at all this year, and it's such a fundamental topic. Or if it isn't that title, we can have a look at title number 1 and see how that concept actually plays into title 1 anyway. So the way to use these titles is, yes, I'm going to go through a plan with you now so you can see how you can link it to those titles. But the most important thing for you is to come up with topics that you are most confident on, thinking about how you could link it to multiple different titles. Because as I said, the chances of it being 100% the same as one of these isn't that likely. but the chances of them being similar to one of these is quite likely. So it's about your ability to tweak it to whatever the exact question title is. So let's have a look at the role and importance of DNA. And I'm going to go through the third one as well. The second one was the prediction that I gave ages ago, and I've already got a video on it. So I'll link that up here. I'm not going to talk through that one again in this video. You can watch that on this previous video. So 1 and 3 are the ones we're going to have a look at. So the role and importance of DNA, a similar title came up relatively recently, and here's an old spec example as well. And these were some of the titles or topic areas that they said would be relevant. Now this did come up many years ago before the spec changed. So don't panic if you're looking at things like DNA hybridization thinking you don't know what that is. That was an old spec topic. But this is just to show you a range of relevant topics that they would have accepted. Now for the essay, you actually only need 4 relevant paragraphs to get into the full marks band. They do recommend that you write 5 just so you have 1 as a backup, but I'm just going to go through 4 today. You could always try one of the extras to be a 5th. So I'm going to start with DNA structure and proteins. So this is where I'd be focusing on the role of DNA naturally. So thinking about talking through protein synthesis. So the fact that the DNA, first of all, unwinds, the hydrogen bonds break because of DNA helicase. One of the DNA strands acts as a template for the complementary mRNA nucleotides to oppositely align, and then RNA polymerase joins those nucleotides together. Then talking through translation, how that mRNA sequence is a sequence of codons, and tRNA will have the anticodons that can align to those due to complementary base pairing, and tRNA brings specific amino acids. The amino acids join together by peptide bonds, which requires energy in the form of ATP. So that would be your AO1, talking through the role of DNA, providing that's code for protein synthesis to create proteins. The importance of then, in this case, would be that DNA is the code. So it is coding for the unique sequence of amino acids, or the primary structure. And that primary structure is what determines the location of the bonds in the tertiary structure. And that is what determines how it folds and the unique shape it has, and therefore the role of proteins. So then we could link it slightly to another topic just by saying that enables either unique binding sites. You could give an example of receptors like insulin receptors, and also unique active sites. So you could talk about an enzyme like Ribisco, for example, catalyzing the Calvin cycle. So that is just a plan for one potential relevant topic. Next option then could be recombinant DNA. So this is now a gene technology's importance of, rather than the natural roles of DNA. So for this one, I'd talk through in vivo cloning. So talk through the process. You could also link it to the fact that this is possible because the genetic code is universal, meaning that the same 3 bases in DNA codes for the same amino acid in nearly all organisms. So for that reason, it's possible for the DNA or a gene from one organism to be inserted into the DNA of another. And you could talk through how that occurs. So restriction endonucleases to cut out a gene, or you could talk about the gene machine or reverse transcriptase. Then talking about how that fragment of DNA or that gene would then be inserted into a plasmid. Again, the plasmid would be cut open using the same restriction endonucleases. And then the DNA would be stuck in using DNA ligase. That recombinant plasmid would then need to be transformed and entering into a bacterial cell. So you get lots of replication of bacteria containing that recombinant plasmid. And you could link this to the human gene for insulin, and that's where I'd get to my importance of. And this is important because it means that diabetics can now be given insulin, which is human insulin. Although it's been created by bacteria, it's based on the human gene. So the protein itself, insulin, is the human version of insulin. So it's less likely to be rejected or to have harmful side effects or nasty side effects. And also it means large quantities can be made very, very rapidly. The next example then is the idea of genetic diversity and the importance of DNA. So we could talk here about how genetic diversity is introduced, and this is where I was saying this particular title, there are overlaps with the third title, which came up in 2020. So maybe it wouldn't be that third title, maybe it'd be more similar to this one, but concepts of variation and genetic diversity could be included in this one. So you could talk about meiosis and or mutations. So for meiosis, we'd be talking about how crossing over in meiosis 1 and independent segregation, both of those processes result in new combinations of alleles, but give a little bit of detail about the process of crossing over and independent segregation. If you're going to talk about mutations, you could talk about point mutations, which are the gene mutations. So that would be your substitution or addition mutations, and talking about how that can result in a new polypeptide chain being coded for, or in other words, a new allele. Then the importance of this, well, mutations results in the creation of new alleles, meiosis creates new combinations of alleles in gametes, but both of those mean there will be genetic variation within a population of the same species. And this is important because it enables natural selection and therefore evolution. So you could then talk briefly about that in this section here. So this means that the species would become more adapted to their environment and more likely to survive. And thinking about the sorts of A-level key terms you could use, so selection pressure, selective advantage, and change in allele frequency, using those terms when you give a very brief overview of natural selection. So the final option I've got here is the idea of the cell cycle and cancer, mainly focusing on cancer. Mutations that are occurring in the tumour suppressor genes or in oncogenes can result in cancer. So you could talk about those two genes. You could also talk about epigenetics. So the fact that hypermethylation of tumour suppressor genes, that can also result in the tumour suppressor gene being switched off. So that would be your A01, talking through those two key concepts, demonstrating your A-level knowledge. The importance of, so that would be then saying the fact that the gene is switched off, or it could be a mutation has occurred, so a different protein is made. That means that the proteins which should be created by those two genes, which control the initiation and the suppression of mitosis, those proteins aren't being made. And therefore, you will have uncontrolled cell division or uncontrolled mitosis, and that results in tumours. And those tumours could be cancerous, therefore break off or metastasize to other organs in the body and cause significant harm. So those are just 4 key ideas of the A01, the A02 that you could include. So the next one then, the third essay, the cause and importance of variation and diversity. So yes, this did come up in 2020. This is the mark scheme from 2020. But as I said, it's such a fundamental topic and it's barely come up. It hasn't been on the advanced information list. It came up on one of the papers as a couple of marks. So maybe this title, maybe a slight variation of this title, or maybe something different altogether, but these concepts could still be relevant, like the DNA example. So we're still going to go through it. These are all of the relevant areas that they said could have come up, but I'm just going to go through 4 again to show you an example of a plan. So the first one is an identical paragraph to the one that we just had on the importance of the DNA essay. So this could be literally the same. We go through first of all the A01, meiosis and mutations, how they are creating the variation. So that is our cause of variation. The A02 is then the importance of that variation, talking about how it enables natural selection and evolution. This is now a completely different concept, and this is linking to immunity, which hasn't come up at all, and it is such a huge topic. So that's why I thought there could be this title, because it can bring in immunity, but also actually the microorganisms essay title, which I've explained in a previous video, that has lots of considerations to immunity as well. So for this one, we could talk through how pathogens' DNA mutates frequently. If a mutation occurs in the gene which codes for the antigen, then the shape of the antigen will change. Any previous immunity to this pathogen, whether that was natural or from a vaccine artificially, it will no longer be effective, as the memory cells will have the memory of the old antigen shape. So that would be your A01, talking through what antigen variability is. The importance of them would be the fact that Because this occurs, it means the virus is able to continue to replicate inside the host until the host's immune system is able to create new antibodies complementary in shape to the new antigen. So you could talk very briefly there about B cells, clonal expansion, plasma cells, memory B cells. You could also refer to the fact that this is why we have to have yearly vaccines and boosters for some infectious diseases. So, for example, the influenza flu vaccine is annual, and you could use a more relevant example of the COVID booster vaccines as well. Next idea is hemoglobin. So within topic 3 for mass transport, it says you need to be aware that there are different versions, there's a variation in types of hemoglobin, so that animals are better suited to their environment. So this fits this title perfectly, this idea of variation and diversity. So the AA1 would be pointing that out. Different animals have different genes coding for different versions of the hemoglobin protein. Then linking it to the knowledge of proteins, because that DNA is different, that results in a different primary structure of the protein, therefore a different tertiary structure. Although, yes, it is a quaternary protein with 4 polypeptide chains, each of those polypeptide chains may be slightly different, therefore fold slightly differently and have a slight different shape. And that is what causes the hemoglobins to have a different affinity for oxygen at different partial pressures. So within that AA1, it's trying to use as many A-level key terms as possible, linking to protein structures, linking to hemoglobins. You could talk about the heme groups, for example. And then the importance of is the fact that this is what enables animals to be better adapted to their environment. And you could give an example. So any animal that is a deep diving animal, so they're going to be underwater for prolonged periods of time, or it could be animals that are at high altitudes where there's a lower partial pressure of oxygen. They will either have myoglobin protein or they'll have versions of hemoglobin which have much higher affinities for oxygen, even at very, very low partial pressures. And that means that they are able to continue to load oxygen even when there's very little there, and they won't unload it until the animal is very, very low and nearly completely out of oxygen. So that means that aerobic respiration can continue for longer periods of time. So the link to respiration would be very brief here, but it would be worth mentioning that oxygen is the final electron acceptor in the electron transport chain, which you would have to write out. I've just said that for short there. And that enables the production of lots of ATP for metabolism. So that's the third paragraph. The final one that I've suggested here is speciation. So you would need to talk through what it is and either go through allopatric or sympatric speciation. I would definitely name them both, but just talk through one of them. That would be sufficient to demonstrate A-level knowledge. So for speciation, that is when members of one species become reproductively isolated and therefore they are unable to have any gene flow, and over many, many generations, they result in individuals that cannot reproduce to make fertile offspring. Or in other words, you've created a new species. You would then talk through an example of either allopatric or sympatric. So whether it is allopatric, which is geographically isolated, you would then have lots of similar concepts to up here. So selective advantage, selection pressure, changing the allele frequency, but that's okay. You are allowed some overlaps like that, because it is still 2 different spec points and different topics. And the importance of, again, whilst there are some similarities here, AQA do state that it is fine to have some similarities in your importance of, because a lot of the time, the importance of is going to be similar, particularly actually this one here, ATP metabolism, that one comes up a lot. But for this one, it's important because it means that disruptive selection occurs. So again, another A-level key term. And we get the creation of a new species. So even when populations become reproductively isolated, each population is able to evolve. So they go through natural selection and they can evolve to their environment, and that helps to ensure the survival of the species, or in this case, creation of 2 new species which can both survive. So that is it. Those are my 2 titles that I've gone through in detail. The third one, you can have a look at the old video. I hope it helps, but remember, the key is knowing how you can link these titles to potentially similar ones that might come up. So don't literally just learn this and do nothing else. It's about knowing how you could tweak it, apply it to slight variations of these titles. So I hope you found that really helpful. B has been happily chilling here the whole time, as usual. Look out for my video that I'm going to be uploading in the next day or two as well, with my 4 final tips of how you can maximise your marks for paper 3. I'm also going to be going live on Instagram and TikTok, so you can check out all of my lives helping you with the exam questions.